chat a little bit about a few concepts from last time. We talked about transcription and translation, right? Remember that? Well, somebody tell me what's transcription. What does that mean? All right, making a complementary RNA, right, based on what? The sense strand or coding strand of the DNA, that's right. And where in the cell does that process of transcription take place? Nucleus, right. All right, now once that messenger RNA, that's what we made, is a complementary messenger RNA, where does it go? Doesn't stay in the nucleus. Where does it go? Goes out and links up the ribosome out to the cytoplasm, and of course the greatest concentration of those is found in that rough endoplasmic reticulum. All right, so now the messenger RNA goes out into the rough endoplasmic reticulum. The two units, subunits of the ribosome attach. They get aligned by way of that ribosomal RNA with the start codon of the messenger positioned at the P side of the ribosome, right? How are you going to recognize the start codon when you see it coming down the screen as broad as life, twice as natural? As the sequence AUG, that's right. And of course, what is the first amino acid that gets inserted for that start codon? Methionine, right. You look there on your codon chart, and there it is. It tells you which amino acid they produce. All right, and then, we're undergoing now what we call, of course, translation, which is basically translating that messenger RNA sequence into an amino acid sequence. Okay? Well, let's take a look at the practice problems, because I think we'd already seen the animation. I think we kind of finished up with that last time, did we not? Yes. See, I haven't totally gone senile. And so anyhow, let's take a look at these, and let's look at the first one. So let's put up here our coding or sense DNA. That's what we've been given, and we'll go from there. Now, I think it's easier when I do this 3 at a time. So let's see. All right, now there it is. Now, complementary to that is the non-coding or non-sense strand of the DNA, which of course is complementary to it, and that's what we were asked to put up next. So we need a volunteer. Ms. Lewis, you're already up on your feet. There you go. Just go up and write that up above it. Oh, you don't really need anything with you for this. No, you can take it if you want. Oh, you already got it done? Excellent.
No, I don't have it all done, but
well,
I can try.
All right. Well, you'll do more than that. You're going to succeed. All right. Now, just write up above it. Just put the non-coding DNA. All right. So, that's off to a good start. Okay, I think that's right. Let me see if I can do something with this. Yeah, that's the problem with that, but I think it's easier to write above. Let's see. I got distracted when I was trying to get a paper clip down here. Let's see. I think that looks right. Yeah. Okay. Got it. All right. Now there it is. Now remember, it's complementary to the coding or sense strand of DNA. So C pairs with G and A with T, and there you go. All right. Now, the next thing we want to put up is the messenger RNA. That would be transcribed from that coding strand of the DNA. Now remember, you use the coding strand of the DNA as the pattern or the template for this. Don't make the mistake that some people do, where they take this as the pattern. This is the one to use, the coding one. So who wants to come up with that one for us? Let's get somebody on this side for you. Bless you. All right, now the thing to remember here, of course, is there's no thymine in RNA. It's uracil instead. Okay, so there we go. Excellent. There you go. All right. Now we've got that. All right. Now, the next thing that we have, now that we've done the transcription, is to do the translation. And this is where we go out to the ribosome and link up with it. So, let's see. Let's get somebody from over here. Come on down.
You're going to show you where it starts?
Yes, you have to show us where to start, and then we go from there. All right. Right. So you're finding the start codon, A to G, and that one's methionine. Okay. Now, the next one, I'll just go ahead and do, we'll say, highlight it the way you did there. Because remember now, the codons are read 3 bases at a time. And I think it's a lot simpler if you just underline it like that. All right, then you got GGG, I think that was glycine. Let's see, where are we? Yeah, it only puts out 3 amino acids. All right, there you go. Because this one, remember, that's a stop. All right, and there's no translation for it. Anything that precedes the start codon, ignored. It's not translated. Anything that comes after the stop codon, forget it. It's not translated either. Just ignore it. All right? Well, one more thing to put up here, and that concerns the anticodon on the transfer RNA. Now, an anticodon, remember, is complementary to the codon. And this is easy because if we didn't use the codon, we're not going to have an anticodon for it. So who wants to come and put those up for us? Oh, let's get somebody over here. Oh. Oh, yes, go on. Go on, we will help you.
Yeah, because I'm not.
All right.
Behind.
You're fine. All right. You don't even need that. Don't even need that. All right, now. All right, now here's the deal. What was the first codon that we translated into an amino acid? AUG. All right, now what would be complementary to AUG?
It would be the opposite.
Yeah, so write it down. U. All right, no, put another here. Then we'll keep it all together. Yes, that's neat and tight.
So I guess it would be
U.
And then it would be A.
Yes.
Okay.
And then it would be.
Opposite of G would be C.
Right, that's it.
Okay.
All right, now, the anticodon, and if we brought up the animation which would have shown this, The anticode on this transfer RNA that's carrying the methionine would have been UAC because that's complementary to the AUG that's on the messenger. All right, well, next one. GGG, write it down. There you go. Now, the next one for the glycine then it's glutamine is
G-U-C.
Yes, that's it.
Okay, G-U-C.
And stop.
Stop.
All right, now why do we not want to go any further? Because remember what happens here. The reason why that's a stop is because there is no transfer RNA that is complementary to it. Okay? So you're done. The only transfer RNA anticodons that you've got to put down are the ones that actually carry amino acids into that ribosome. That's it. If they didn't bring an amino acid, forget them. They're not applicable. All right? Now see how easy that is, isn't it?
Yeah.
Heavens, yes. Well, we've got another one. Let's take a look at it. Okay. All right. So, everybody's got all this stuff, right?
Yeah.
Okay. Well, let's put the next one up, and we'll do it the same way. All right, so I'll just get rid of all this. All right, and we'll just put up the next one. And let's see, it goes GTT, TCT, TAC, and CCC. All right, there it is. All right, now we need somebody to come up and put up, since that is the sense or the coding strand of the DNA, somebody to come and do the antisense, nonsense, non-coding, whatever you want to call it, DNA. There it is. All right. Everybody agree with that? Looks good to me. All right. Now, next one. Somebody put up the messenger RNA that would have been transcribed from the coding strand of the DNA. Let's see. Somebody that hasn't been up yet on the side of the room. Oh, there you go. All right. Let me see, is it? Let me check this other blue marker. Maybe that one's about dead. I don't know. Try this and see if it's darker. I don't know. Yeah. Okay, I think that gets it. Okay, very good. All right, next, we need somebody to come up and translate for us. So let's see here. Have you got it worked out?
Yeah.
Okay, just bring that with you then. All right, so there you go. You find the start codon and start translating it into an amino acid sequence. All right, so I'm going to find histidine, leucine, and that's a stop. All right, so that's it. So again, we only have 3 amino acids this time. Okay, now somebody can come up and put up the transfer RNA anticodons for us. Okay, here, why don't you go up and do that? You can do it. All right, now here's all you got to remember. The only ones you're writing down are anticodons for transfer RNAs that actually brought amino acids to the party. So if it didn't bring an amino acid, you don't write it. So you only have 3 of them right. All right, so here's the first one. That brought in the methionine. There's the codon. What would be the anticodon sequence? Just write it right under there. All right, what pairs with A on RNA? Not T, U for RNA. Okay, because remember, this is RNA. So the U, all right. Now what goes with uracil? A. And what goes with guanine? C for cytosine. There you go. All right, now that's it. Now that's done. Now the next anticodon for the next transfer RNA would be G, U, G, right? Cytosine and guanine pair. All right, now the one that brought in the leucine, what would be the anticodon on it? Yeah, GAA. All right, there you go. It's just complementary to the codon. All right, no, don't go any further. Stop right there. Why? What did we say about stop codons? There's no transfer RNA complementary to a stop codon. You're done. That's it. All right? Well, that's all there is to it. Now, do you think you could do something like that for me?
Yeah.
Well, you know, next Tuesday when we have our little written exercise, you're going to get the chance to do it. I'll be more than glad to give you a codon chart to refer to. And I would expect the problem to be very similar to this. Now, what about this? Just a few steps, but it's easy. It's worth a lot of points. You can rack up some money in the bank with something like this. Okay? Providing you don't make a dumb mistake. Yeah, okay? And here's the things you gotta watch. I'll ask for the non-coding DNA, and you'll find people will use that as the template to make the messenger. You know what they've done when they do that? Red ink comes out and flows all over their blue book. Remember, the coding strand is the one you use to transcribe to make the messenger. Alright? That's the one. So keep that in mind, and you're not going to go wrong. You can't. It's too simple. Really. Alright? Alright.
When is our test again?
Next Tuesday. Yes, and what's on it? Well, it starts with cells, organelles, cell cycle, metabolism, it's part of metabolism, and then we have the integumentary system, which we're going to start today and finish up Thursday. So we'll be all ready for that early visit from the Easter Bunny next Tuesday. Okay? Simple enough, isn't it? Yes. Alright, so everybody got all this stuff? Alright, well, let's... Alright. So, let's get to the integumentary system. Alright, well, let's take a look at this. Let's see if the thing's working. It probably went to sleep. Alright, now let's see if it'll pop up. There it is. Alright, very good. away we go. All right, the integumentary system. And what it is, we know, is the skin and its appendages. Well, what are the appendages? Well, hair and nails would be the most obvious, but there are a few others. We also have glands, and there are glands in the skin, and receptors. Now, you all remember the old homeostatic mechanism, right? We have receptors, right, that receive stimuli. Where do they relay that information? Central nervous system, right, for interpretation, against the set point. If things were out of balance, what did they do? Generated an impulse that went to a effector. And what kinds of things served as effectors? Muscles and glands, and we're going to have both of them in the skin. All right? All right, now that's simple, isn't it? All right, skin, biggest organ in the body. Remember, that was a question on the first exam. All right, how much is it? Don't worry about these numbers. About 19,000 square centimeters, or about 3,000 square inches. Now, I wonder how that was ever determined. Well, you can guess. Human history is full of all kinds of gross things. I mean, like the French chopping heads off with the guillotine to see how much blood, they drained blood out of them. Somebody up here did drain it out and see how much is there. All right, and how much, how much volume, all that stuff. Well, there you go. Now, there's other ways to do it. I mean, you could lay somebody down on a sheet of paper, and you could trace around them on all the surfaces and put a grid on it and calculate that way. That'd be too simple and easy. All right, thickness, anywhere from about 0.5 millimeter to 3 millimeters. It's not very thick. And the thicker areas are going to be on the areas that get the most abrasion, the most use, like the palms of the hands, soles of the feet. And as we also note, the dorsal surface is probably thicker than the ventral surface. Well, what about the dorsal surface? What is the dorsal surface? The back, right. Well, where do you spend your time sitting in your chair? On the dorsal or ventral surface? The dorsal surface. Well, what about when you're in bed? Probably most of it's on your dorsal surface, although, I roll around a lot, and probably a lot of you do too. But anyhow, there you go. What does it do? Covers the body, protects the underlying tissues from lots of things. Protects them from infection, for example. The skin is a mechanical barrier. There are very few organisms that can actually penetrate the skin. There are some that can, but it's not very many. But if you didn't have any skin there, anything could get in. Anything. All right? Now, keeps you from drying out, prevents dehydration. Well, now, what happens if you peel off your skin? What's underneath there? Muscle. It's probably muscle right under there, right? All right, and that's very moist, normally. But what would happen if you took the skin away and you didn't do anything to protect it? It would dry out, wouldn't it? Like our poor little rats and piggies, when we're done with them and we pop them in the hood and let them go overnight, and they come back and they're all dried out and mummified and ready to go off to their plastic bag sarcophagus to head for the landfill. All right. Also, radiation. Ultraviolet radiation. From the sun, constantly exposed to it when you're outside. Well, yeah, it can damage the skin, but it can be more damaging to the underlying tissues. So the skin serves as a barrier to help protect those. All right. It helps to maintain temperature, and we'll talk more about that probably Thursday. But in any event, as you well know, what happens when you're really hot, like you can get sometimes in this room because we get a little bit warm? You set and sweat or perspire. All right, why do you do that? That's to help cool you down. What happens when you get cold? Well, yeah, but shivering's not part of the skin, but goosebumps are. All right, well, that's another modification because that cuts down on surface area and you don't radiate away as much heat. More on that later. Prevents the loss of nutrients. Well, where are the nutrients? Where do the nutrients go into the cells? The muscle cells, for example. They're in tissue fluid, right? You've got salts, you've got glucose, you've got amino acids in tissue fluid that came from the blood. All right, if you didn't have the skin, what's going to happen to that stuff? It's going to drip out. You know, the thing is, you've got someone who's been burned and their skin is no longer intact. You've got to take precautions to prevent the loss of nutrients, and therapy, IV therapy, for example, you know, put back what they're losing. All right? Excretes water, salts, and waste products. All right? That's one way to get rid of waste. What's the major way you get rid of water, salts, and things like urea? Urine. That's the major way. But we can get rid of it some this way. All right? Through perspiration primarily. All right? Absorbs compounds. Now the deal with this is it probably better be something non-polar, because things that are water soluble, water is not really absorbed well through the skin. The skin waterproofs. That keeps water out, and it also keeps the water in that's down here in these underlying tissues. All right? Receive stimuli from the environment. Touch, pressure, pain, things like that. All right, we're going to have receptors in the skin that, in essence, serve to detect those stimuli. All right, and synthesizes vitamin D. Where do we get vitamin D? Well, from cholesterol. Cholesterol exposed to sunlight, again, UV radiation helps out here. And you end up with vitamin D. Why do you need vitamin D? Vitamin D is necessary for the absorption of dietary calcium. So if you want to absorb calcium from the diet, you've got to have some vitamin D. Now, this time of year, we're not out in the sun that much. You can end up with prison pallor, as you could call it, being pale and all that sort of thing. Well, probably not making a lot of vitamin D. Although, you know, at the latitude where we are, that's usually not that big of a problem, but it can be. So you may have to have supplements for it. But in the summer, if you're out in the sun to any extent at all, you're going to get enough sunlight exposure to make adequate levels of vitamin D. The thought is, of course, a lot of people are probably deficient in it, but you know how it goes. Sooner or later, you're deficient in everything. You know how it is, you know, or something's bad for you or good for you, whatever. All right, the structure. There are three basic layers to the skin. And the first one is called the epidermis. Now, if we get out our terminological dissecting kit, what does the prefix epi mean? It means over. Over the dermis, literally. Now, I'll bet having said that, you couldn't guess in a million years, if you had that long to think about it, what's the second layer going to be? The dermis, right, you got it. All right, now what about it? Well, the epidermis turns out to be the thinnest, most superficial layer. What is it? It is essentially a keratinized, stratified, squamous epithelium. That's what it is. Keratinized, stratified, squamous epithelium. Now, epithelial tissues. What was their function? What was the function of epithelial tissues? Covering and lining, right? That was their primary function. All right, well, I think we'd agree the epidermis is covering something, isn't it? All right, remember that epithelial tissues can come in the forms of either being simple or stratified, right? What was the difference?
Simple is one layer.
One cell thick, that's right. And every cell touches the basement membrane. Stratified means it's multiple layers thick, right? All right, so we have multiple layers here. Only the very bottom or basilar layer touches the basement membrane. What's the basement membrane made of in an epithelial tissue?
Polysaccharide.
It's a polysaccharide, that's right. and those basilar layer of cells actually make it secreted. All right? Now, if we're talking about squamous epithelium, what does that mean? Thin, flat cells, right? Remember, squamous comes from scale, okay? All right, so there they are. They're a thin, flat cell. The top layers are the thin flats. The bottom ones tend to be more cuboidal or columnar in shape because they flatten out as they rise up, okay? And that's going to be true here. All right, second layer, one right under the epidermis, dermis. Thicker, consists largely of connective tissue. It's going to have some other things in it, like blood vessels and nerves, all right? And receptors and hair follicles and glands, so there's a lot more down here. Now, one other thing to remember about epithelial tissues are avascular. What does that mean? No direct blood supply, right. The blood is down here in the dermis. So the nutrients are going to have to diffuse out to those cells of the epidermis. And as you might guess, the ones that are down near the bottom, that are closest to the dermis, are the ones that get their lion's share of the goodies. And the ones that are a few layers above, well, poor things, lived a good life, and now they can starve to death. All right, so there you go. All right, and then we come to the third layer, which isn't terribly thick. It's called the subcutaneous tissues. And another term for the hypodermis. Well, hypo simply means below or under. So if it's hypodermis, where is it going to be? It's under the dermis, obviously. And if you have a hypodermic injection, where does it go? Under the skin. All right, there you go. All right, what do we got here? Areolar connective tissue and adipose. Oh, lovely. What's areolar connective tissue? Sometimes called loose connective tissue. What kind of fibers did you find in it? Remember that from lab? Elastic fibers, right. Collagen fibers, yes. One more. Reticular fibers, bless you. That's it. And remember now, they're not densely packed together. There's a lot of open space in here. All right, and then adipose. Wonder what the purpose of that would be. Well, it does store fat for energy, yeah, but more importantly perhaps here, padding, protection, and insulation. All right, because you want to keep that layer of fat right under the skin. Okay? All right, there you go. All right, because it's there to help pad, protect, and insulate. And that helps also with maintaining the core body temperature. All right, now, the epidermis. The epidermis has about 5 layers that we can pick out. Now, they're not always that evident, and they're and they're going to be more commonly seen, all 5 of them. In thick skin, when you get to thin skin, this top layer, this stratum corneum, you may not see it. It may be inapparent and virtually not even there, really. It's the most superficial layer, stratum corneum. Stratum means layer, all right? And corneum would refer to, kind of hardened-like material, all right? Now, what about it? Well, it can be several layers of cells thick. 25 to 30, I wouldn't worry about that. It's just several layers thick. And these cells are flat, and they're dead. And they're filled with keratin. Keratin is a water-insoluble protein. And not only is keratin, but its precursor substances as well are water-insoluble proteins. So the keratin helps waterproof. Remember, that was one of the functions of skin, waterproofing. Well, keratin and its precursors are going to be responsible for that. All right. Those cells, as we know, are constantly being shed. Constantly being shed. I'm just shedding these as I walk around this room. They're dripping off of me. And in fact, if we would get, a nice, powerful vacuum cleaner and sweep this room, we would find gobs and gobs of old, dead, shed, keratinized cells in everybody that ever walked through this room. You wash these off in the shower in the morning, you know, you shed them all during the day, you know, so there you go. All right, far more pronounced on the palms of the hands, soles of the feet, dorsal parts, you know, of the body. It's the thick skin areas are where you really find this. Get into thin skin areas and it's pretty much inapparent. You may not even see it. All right? Now, peel that off, we get to the next layer and we have the stratum lucidum, the clear layer. Clear layer. All right, stratum lucidum. These cells are also flat and dead. The only thing is, instead of being filled with keratin, they're filled with a precursor of keratin called ileidin. Well, it's also water insoluble, so it helps waterproof as well. All right, and once again, probably thicker in the thick skin areas, but you'll probably find at least a few cells thick of this just about anywhere you look. All right, next one down. The stratum granulosum. Granulosum meaning granular. Now, cells very often take on a granular appearance when they are in their death throes. All right, these are not all yet dead, but they're probably in the process of dying. And what's happening is when you look at their nuclei, the chromatin in the nucleus just condenses down into a big dark staining blob. And that kind of gives them that granular appearance. All right? They are filled up, their cytoplasm, with a protein called keratoilin, and that's also a precursor of keratin. So we have like keratoilin, and then ileidin, and then keratin. You know, it's kind of like in that way. So there they are, the cells in various stages of death and degeneration. All right, now why are they dying off? Well, now remember now, we're in the upper layers. These are some distance away from that basal layer. We haven't quite got down to that yet. And the basal layer is where the best supply of nutrients are going to be. So when you start getting further away from the nutrients, well, what's going to happen to the poor things? They're going to die. They're going to starve to death. All right. Now, next layer, stratum spinosum, the spiny layer. The cells have kind of a spiny appearance to them. All right, now, what about these? Well, these are alive. These are alive, and they have, and here's something we want to put a star by. Within these cells of the stratum spinosum, there will periodically be things called Merkel's discs. And Merkel's discs are specialized endings, if you will, to nerve fibers. They're receptors. They are receptors. Now, what kind of a signal do they receive? Fine touch. Fine touch is what Merkel's discs are for. And Merkel's discs are the only receptor that you find in the epidermis. The others are going to be in lower layers, like the dermis. All right? So the only nerve receptors, the only nerve endings you find in the epidermis are the Merkel's discs. They are for fine touch. Now, where do you think you're going to find more of these? Like fingertips, right? Typically hairless areas more often than anywhere else. And fingertips are probably where you find probably one of the greatest concentrations of these. Because what do you notice with your fingertips? They're very sensitive. I mean, I'm just barely touching that paper and I can feel it. Okay? It's really pressure on the thing is really what's happening. And it just takes very, very light pressure there for those to register an impulse, which is interpreted as touch, fine touch. All right? There you go. Now, the last layer, the innermost layer, the bottom layer, if you will, the stratum bacilli, the basilar layer. These are the cells that actually touch or make contact with the basement membrane. They are also the cells that divide to form new skin cells. So these are the cells that are undergoing cell division constantly. And I wonder what kind of cell division they would use. Would it be mitosis or meiosis, I wonder? I think mitosis is a good one. Yes, why? How many chromosomes are these going to have? 46. They're somatic cells. And somatic cells replicate by mitosis, mitotic division. Right. So they're diploid cells. All right? So they form new cells. Where the new cells go, they stack up on top of the old ones. And over time, what happens? They just get pushed up to the surface. And as they get pushed up, what happens to them? They flatten out. They get thinner. They get further away from the nutrients. They start to die and degenerate. And ultimately, they end up on top. And that stratum corneum is flat, dead, thin cells. They're just filled with keratin. And there you go. Now, the cells themselves, down this basilar layer, are typically more cuboidal or columnar in shape, as we'll see here in a bit when we look at some photographs of these things. All right, and there we go. All right, so sometimes you'll hear this stratum germinativum, which is really just a combination of the bottom two layers, the stratum bacilli and the stratum spinosum. And those are the ones where the new cells are formed and where the cells are very much still alive. All right, so there we go. All right, now let's take a look at this photograph and let's cut the lights for it. All right, so what we have in A there is actually just a drawing. And in B is an actual photomicrograph. And we'll get a bigger one here in a bit. But in any event, here's the epidermis from here out. And the same deal here, like from there out or there out. And you see how it has this kind of zigzaggy appearance to it. And we'll have more to say about why we have that in a bit. But in any event, here is that stratum bacilli. And you see how, well, I don't know if you can see it that well or not. This is in your book. But those cells actually look more columnar than anything else. And the basement membrane is just right along here. All right, and then we have, like the next layer up, the stratum spinosum, and then we have the stratum granulosum, where they're starting to die off, and then right up here would be like the stratum lucidum. Those are dead and flat, and they're filled with, in essence, eleidin. And then up here is the stratum corneum, and those are the old dead ones. And a lot of times, when you have skin that's been fixed and sectioned, that stratum corneum wants to peel off and separate like that. Because remember, those are dead, so there's not a lot to hold them there. And, those are the ones that you're constantly shedding off anyway. Down here, we're looking at the dermis. And so it's down in here where you're going to start finding capillaries and lots of nerves and, and so on. The glands, the hair follicles, muscles, and so on. All right, if we go to this one. Now, this is obviously from thick skin because from here up, that is the stratum corneum. All right, so all of that is quite dead. And in this case, it isn't separated off like it was in the prior slide. And then, then you can pick out some of the other areas, like the stratum lucidum and the stratum granulosum, and then down in here we've got the stratum spinosum, and then all the way down to the very bottom, the stratum bacillum. And then here, dermis. And you see this projection of dermal tissue up into the epidermal area called the dermal papilla. Well, more on that one here in just a bit. All right? So, there we go. Now let's chat a little about the dermis, the next layer down. Now this one's a bit thicker than the epidermis. And, you probably have actually peeled some of your epidermis off. Did you ever, I don't know, like scrape the back of your hand on some sharp object and you notice some little like whitish material rolls up? Well, that's the epidermis. You're peeling off some of it. It doesn't feel too good. You go. All right, connective tissue, collagen fibers, elastic fibers. All right, blood vessels, capillary, and small blood vessels, capillaries, venules, arterioles, yes. Nerves, glands, hair follicles. All right, more things showing up there in the dermis. Primarily connective tissue, but we also have a blood supply there to handle, of course, not only the dermis, but to nourish the epidermis as well. Because remember, the epidermis is epithelial tissue, which is avascular. All right, now, there are two regions to this dermis. There's an upper and a lower region. The upper region is called the papillary region. Papillary. All right, and it's in that papillary region where those finger-like projections called dermal papillae are found. Now, we just saw that on the slide. Let's back it up. There it is. There's one, there's another one. All right? Finger-like projections of the dermis up into the epidermis. Those are called dermal papillae. And what about them? They lead to the epidermal ridges, or the wrinkling of the epidermis. And what do you call these wrinkly things that you see here? Fingerprints, palm prints, toe prints, footprints, right? Yes. Now why have we got them? They increase friction for gripping. All right? You know, back in the old days, and of course, I mean, some people like to still go barefoot in the summer, particularly, well, you know, climbing a rock, climbing a tree, or whatever, you know, you got extra grip. When you go to open a jar, how do you do it? You know, you want the grip, you want the added friction, right? You don't stick it here and go like that, right? I don't. All right? Right? Probably have a lot more grip here than would here. All right, there you go. All right, so, and if we go back to this slide for just a minute, another little thing we can point out. You notice, I don't know how well this shows up, we don't cut the lights. You see how this is wrinkled up here? You know, you can have a print up there as a result of that ridge. All right, there you go. All right, so there we go. Now, another nerve ending, Meissner's corpuscles. These are nerve endings. Where are they found? They're in the papillary region of the dermis. Papillary region of the dermis. Therefore, touch as well. What's the difference? What's the difference between these Meissner's corpuscles and the Merkel's discs that we mentioned earlier? What were Merkel's discs for? Fine touch. These are for just touch. Well, they're deeper. You're going to have to press harder for those to be stimulated. Okay? There you go. Well, that's simple, isn't it? Heavens, yes. You know, what if someone who's sight impaired and they're using Braille? How are they reading? They're touching the page. There are those, bumps or raised spots on there that they're feeling, all right, the pattern on. Well, it's a combination really of the Merkel's discs and Meissner's corpuscles that they would be using and relying on. And, a lot of times, like if you're sight impaired, well, some of the other senses like touch would be heightened a bit. All right, so there you go. All right. Now, the reticular region of the dermis, that's the lower region. This is going to have many of the same things, but it's mostly a dense, irregular connective tissue. So now we just have like bundles of collagen fibers, bundles of elastic fibers, and again, blood vessels are there, and nerves, and so on, and adipose, a little bit of adipose, of course. But in any event, there you are. It's just more connective tissue than really anything else. But, the other things are certainly there as well. All right. And glands start to show up here. That's something else I need to mention. We'll chat more about these here in just a bit. Glands start to show up down in this reticular region. Sebaceous glands, those are the ones that produce the oil. Ducts of sweat glands are going to be there, all right? And sudoriferous glands are sweat glands that produce sweat or perspiration, if you want to be polite. You know, and there you go. All right, more on those in a bit. In fact, we're ready for sebaceous glands now. There's a few tidbits we want to point out here. What are they? Simple branched center holocrine glands. Oh, let's dissect it, shall we? Now, first of all, a center. That refers to the term acinus, A-C-I-N-U-S. Okay? And acinus, A-C-I-N-U-S. I'm going to spell it rather than pulling up the screen to write it on the board. What's an acinus? A ball of cells. A ball of cells. All right? Like a sphere. Now, glands. Glands are actually made of modified, specialized epithelial cells. All right? Epithelial cells that are specialized to produce some kind of product. All right. Simple branched. Well, branched little balls, okay, going into some kind of a duct. Now, holocrine. That's a nice, important little term. I like to think of holocrine as whole cell. Whole or entire cell, all right? Now, here's why. How do holocrine glands secrete their product? Well, the product is made and accumulates in the cytoplasm of the gland cells. And then to release it, each cell breaks off, and when you break a cell off from its neighbors, what's going to happen to it? It starves to death, it dies, and it breaks apart. And when it breaks apart, it releases its product, namely sebum, or the oil. All right? So, in other words, if it's a holocrine type gland, it releases its product when the cells die and break down. All right? That's how it's released. Now, sebum, it's an oil type stuff. Lipid, right? I wonder what kind of organelle these cells would likely be very well endowed with if they're going to be producing an oily-like material as their principal product. Where were fats made? What kind of organelle was responsible for making and storing fats? Well, I'll give you a hint. It's a type of endoplasmic reticulum, and it's not rough. Smooth endoplasmic reticulum, right! All right, so as these cells carry out their normal existence, they're producing a lot of this sebum. It's just collecting in their smooth endoplasmic reticulum. So when it's time to release it, what happens? The cell breaks off from the neighbors. It runs into the duct of the gland. It dies. It breaks down. As it breaks apart, the sebum comes spilling out into the duct, and there it is. There it is. That's a holocrine gland. And there are going to be other examples of holocrine glands. All right? Now that's simple, isn't it? Milk is produced by holocrine glands. All right. There are other comments. All right, so there we go. Now, what is this sebum? It's an oil. It's a mixture primarily of fatty acids, cholesterol. There's some proteins, there's some inorganic salts, but there's very little, if any, water to it. It's a lipid material. Why would you have it? What good does it do for you? Well, primarily it keeps your skin moist and pliable and keeps your hair from becoming dry and brittle. I don't have much to keep, but anyhow, there it is. All right? And, of course, in the wintertime, when you're not producing enough of it, it's how your hands can crack and all that kind of stuff. Well, there you go. Most of these sebaceous glands are actually associated with hair follicles, as we'll see here in a bit. And so, they typically secretes their product into the hair follicle, and the sebum then gets out to the surface of the skin along with the shaft of hair. And we'll talk more about what hair is a little later. We'll get to that Thursday. All right, now there are going to be areas of the body that are hairless, obviously. We need sebaceous glands there. There they actually have ducts that rise up to the surface and allow the sebum to reach the surface of the skin through the ducts. All right, now here we have another little diagram. This really shows all three layers. There's the epidermis. Here's the dermis. Down here is the subcutaneous tissue. All right, we haven't talked about it yet, but as you might guess, it's largely going to be areolar tissue and adipose. All right, here we've got, we've got sweat glands, we've got nerves, nerve endings, hair follicles. There's an erector pili muscle. More on that one in a bit. All right, blood vessels all over the place. There you go. A lot of action down in that dermis, even though it's kind of, doesn't sound real exciting. And, of course, what does this subcutaneous layer do? It basically connects all of that to the underlying tissues. And what was the purpose of all that adipose and the subcutaneous tissues? Protection, padding, and insulation. Right. Okay. Now, the subcutaneous layers, we've already said, areolar tissue and adipose, anchors the skin to the underlying tissues. If we do rats in lab, first thing we're going to do is skin the poor little things. When you're ripping their fur off, what are you doing? You're ripping that areolar tissue. They're dead. Don't worry about
Really
it.
nice
Poor things. Rats don't get a lot of sympathy. I know it's cruel, but anyhow. Now, another type of nerve ending, and this you find in subcutaneous tissues, and that's the Pacinian corpuscles. And they are pressure sensitive. They are there to sense pressure. Well, what's pressure? You're just pressing harder, right? That's really the difference between touch and pressure, is the force. Pressure is the pressure. Well, makes sense, doesn't it? That can be deeper in the skin than the ones that you have, like the Merkel's discs with fine touch. Right? Yeah. All right, so there you go. All right, something else down here, sudoriferous glands. Those are the sweat glands. All right? Now, as I told the other class, use that word in dinner conversation to impress everybody how much you learned today. Sudoriferous glands, most people aren't going to have any idea what that is. And there's a modification on it called ceruminous gland. All right, sudoriferous glands, coiled or branched, tubular, apocrine glands. Oh my word, a few more terms. Coiled, what would that mean? Yeah, kind of in a whirl. All right, tubular, just a tube. All right, it's just a tube. All right, just line the cells. All right, and the material goes in the center of that tube, the duct, and gets taken out to the surface. Apocrine. All right, now that's the second type. There are actually three types of glands based on how they secrete. All right, apocrines, and the key word here is apex. A-P-E-X. Apex. What is an apex? Well, it's not always a top, but it's a point. And for this, it is the top. All right? All right, point. And it is the top in this case. Now, what about apocrine type gland? Well, the product is made in the cell, and it accumulates in the apex of the cell, the top point of the cell. All right? Now, when it's time to release the product, the apex of the cell breaks off. Just the apex, not the whole cell. Just the apex. Well, what happens to that part that broke off? Dies, degenerates, breaks down, releases the product. That's how the product gets out. Now, we didn't get rid of the whole cell this time, like a Holderman gland, right? So what happens with the rest of the cell that remains behind? Well, it grows a new apex and fills it up with stuff, and then it breaks that one off and sends it on its merry way. So the cell hangs around longer. You know, if you've got Holderman-type glands, you've got to make lots and lots and lots of cells because you're constantly losing them. Well, you don't lose as many with an apocrine-type gland. Oh yeah, you lose them eventually, because no cell's going to last forever. But, you know, they hang around for a lot longer, and they produce many, many apices, I guess we could say, and, you know, put them out, and there you go. Now, what about perspiration? Well, this is water-based, not oil-based, like sebum was. So, it's mostly water. Now, what else have we got in there? Well, we've got salts, like sodium chloride, all right? And then we got lots of things that will dissolve in water, like the waste products, urea and uric acid. You want to get rid of those, and you get rid of them with any body fluid, really. Amino acids, well, those are nice to hang on to, but we do get rid of some of them this way. Ammonia, and who wants to keep that around? Glucose and lactic acid and ascorbic acid, they're nice to have around too. They're not really waste, but there's going to be a little bit of them present in this perspiration, because, I mean, really, when you go filtering the blood, and essentially that's what you're going to have these kinds of things present in the product. And there it is. It's a watery-based material. All right, and it gets out to the skin primarily through ducts. All right, now when it comes to perspiration, as we know, it helps to regulate temperature. It's a way to get rid of waste, but it's not the major way to get rid of waste, as we've already said. Two kinds of sweat glands, or sudoriferous glands. Apocrine sweat glands and eccrine sweat glands. Now, which are not as widely distributed? The first one. A comes before E, so I like to remember. That's not what I remember. Apocrines have a limited distribution. Ecrines, they're more often spread all over the body. All right? So let's take a look at that. Apocrine sweat glands, simple branch tubular glands, I wouldn't worry about that. Here's what I put the star by. Where do you find them? Axilla, pubic region, pigmented areas of the breasts. Limited distribution. Limited distribution. Axillary area, pubic region, pigmented areas of the breasts. Axilla, where's that? Under your arms, that's right. Those are apocrine sweat glands. All right? That's simple. All right, secretory portion in the dermis, ducts opening into hair follicles. All right, so material comes out primarily through the hair shaft, or with the hair shaft, right beside it. Kind of like what happens with sebum, really. All right, now, okay, we already said that. Come on, next. Ecrines, simple coiled tubular glands. Wouldn't worry about that, where you find them everywhere else. In fact, what we note here is where you don't find them. They're not at the margins of the lips. Did you ever hear anyone with sweaty lips? No, I never did either. What about sweaty fingernails? Nah. All right, things dealing with the genitalia and the eardrums. There's no kind there that we'll get to that doesn't produce sweat. All right, there we go. Now that's simple. Ecrine sweat glands. All right, these secrete through ducts that rise up through the dermis, up to the epidermis. They're not associated with hair follicles. All right, so which kind of sweat gland is associated primarily with hair follicles? Apocrine sweat glands, right? Yes. I'll see that. All right. Now, before we get to skin color, in fact, that's where we're going to pick up next time, there's one more kind of sweat gland, a modified sweat gland, that we want to mention. And it's a ceruminous gland. And it produces something called cerumen. That's earwax. Where are you going to find ceruminous glands? Well, yeah, the auditory canal, all right, and it's a waxy-like material, and really these are modified sweat glands, are really what they are, even though they produce a waxy-like material rather than a watery-based, fluid type material like down in perspiration. Okay? Now that's simple, isn't it? Heavens, yes. So these have the least, common distribution, I guess you could say, of any of the sudoriferous glands, because there are modified sudoriferous glands. All right. Well, I think wait a minute. Excuse me, I'm not counting on for spelling today. It's an I. Well, there we go. Now see, I've scarred you for life because you'll think it's an E. But anyhow, there you go. All right, we'll pick up with skin color the next time, and we'll finish off the skin. There's not a lot more to go. And then we'll be ready for next Tuesday's exam. And as I said, cells, organelles, cell cycle, metabolism, the DNA problems, of course, included there, and the integumentary system. I think that'll keep you out of mischief. Okay, very good. Okay, we can make arrangements on the test. Okay, see you later. Bye.